Hi, how are you, Sounak? Thank you for making it.
Good, how are you doing?
I'm good too, thank you. Let's wait like a few more minutes to let people arrive and then I'll introduce you and then we'll start. Thank you.
Sounds good, thank you.
Okay. Yeah, we can slowly start and then we can always refresh the room. So hi Abyss, how are you doing?
Hey, Kat. Hi, Solmac.
Hi.
Okay, so welcome everyone to the Science Society Club today. We have our guest speaker, um, Sounak Mohanta, who will talk about his research and Dr. Salman's lab. And yeah, it's a really interesting study and I think we will all learn a lot. But let me first introduce you a little bit. Um, so Sunak, he is, as I said, a graduate student in the Department of Psychology in the Salman Lab, and he is interested in predictive coding and how subcortical activity helps us integrate information from different modalities as well as perform sensory motor transformations. He uses neuroimaging and multi-site electrophysiology in non-human primates and human subjects. And yeah, he published, like he's a PhD student, but he has so many really great publications in high impact journals. So I congratulations to that. And yeah, thank you so much for taking the time and welcome to our club. So thank you. The stage is yours.
Thank you, Katerina. Thank you for the invitation also. So, The story I'm going to tell today is about how we make predictions about environment around us, or predictions about what is going to happen next. So as you can think of it, it is something we do quite regularly, every day while walking around. But as neuroscientists, it has been quite overlooked too, also. So because traditionally we have interpreted brain's processing strategy as pretty much bottom up, so building from the basics, taking small chunks of information and then processing them separately, and then finally joining them together to build a bigger concept. So let me give you an example so that it's easier to understand. If I show you a picture of the car in this framework, what your brain will be doing is probably taking first the sub parts, so the wheels, the steering, the seats, processing them separately, and then in the next step putting them all together to create um, perception of a car. And the brain will be doing this each time from scratch whenever you see a car. But if we think more about this, it seems quite inefficient, right? So even if I have seen a car before, it really doesn't matter, because even after that, when I'm given a second for a second time, I'm shown a car, it will start processing from the scratch. But what I really want to do, if I think about a more efficient strategy, is that the moment I see a hint of four wheels, maybe a bit of the steering, I should bring back my previous experience of how a car looks like, and then very quickly have a quick perception of, oh, this is a car. So in last decade or so, there has been some change among the cognitive neuroscientists about how perception happens, and a recent idea called predictive coding, though recent in the history of neuroscience, but actually if we look back, it is quite rooted in 19th century Western philosophy and also in certain schools of Indian philosophy. So this framework has actually flipped this idea of perception on its head, and instead of processing every bit of information as if it's new and as if it has never seen it before, scientists have suggested that our brain actually builds a model of the world around us, and then what it does, it checks every incoming information against that model. So if the incoming new information is matching the model, it just well and good, it just keeps the model as it is. But otherwise, if there is a mismatch between the model and the new information, it would incorporate those mismatches to update the new model so that it better represents the world. So even if we start with a very naive model of the world, and then in multiple iterations, if it can update and incorporate the mismatches, at the end we'll have a model that correctly represents the world around us. So let me elaborate with another example. So we all have this model of a model of our kitchen in our brain, right? So we know where the toaster is, where the burners are, how does the kitchen top look like, and uh, um, and we know where, say, I'm a coffee addict, so I know where I keep my coffee mug. So for say, one morning I just walk into my kitchen just looking for a coffee mug, and I know that I have kept it last time, or my first prediction or anticipation is that it is next to the toaster. But when I look for it, it's not there. There is this mismatch between where my existing model of my kitchen predicted the coffee mug to be and the, where I try to find it and it's not there. And then I look around and it's somewhere else. I had moved it probably. But then I use this new mismatch information to really update the existing model of my kitchen, so that in the next iteration, in next morning when I walk into the kitchen, I exactly know where the coffee mug is, right? So that the new model replicates the correct map of the kitchen. So in summary, what the brain is doing in this framework is it has a three-step procedure. It constantly first makes a prediction, and then it checks uh, then it checks if the prediction and the whatever my surrounding is matches or not, and if it doesn't match, then it updates and does this over and over again when we are moving around in our daily lives. So building on that premise, what we wanted to do in this study is test do have two aims specifically. First is, if this kind of predictive processing is really true, what are how does it help us? How does it make our decision making more efficient? And the number two is, if it is really true again, but if it is really true, what brain signals exactly correlate or replicate, reflect this kind of processing? What brain signals, what brain connections? So the experiment we designed is quite straightforward. We designed a very boring computer video game, and what the participants have to do was tell us that if a sound and an image came from the same pair or they are from a different pair. So for example, if I show you, if I present you sound of a cat meowing, and then followed by that an image of a cat, you will, because they're associated, you will report that okay, they are paired. On the other hand, instead of a cat's picture, if it was a dog's picture, then because it's they are not associated, it's a different association, right? So in that case, you will report that okay, they are not paired. So conceptually, our boring video game was exactly like this, and the only difference we had was that first we had three sounds, and each sound was paired to an image. So three pairs of sound and images. And also the other critical thing was the all the sounds and all the images were novel. So this is not something that we encounter in daily life. So the sounds were nonsense English words, and the images looked like animals, but they were not really any pictures of animals. And we taught the participants these associations, so associations between each sound and each image. And then once they learned the associations, what we did is skew the predictive qualities of the sounds. Um, so what we made is we made certain sounds highly predictive of its paired image. So for example, when you say hear sound 1, you will see its paired image for 85% of the times. So very highly predictive. And for another sound, what we did is we made it moderately predictive. So when you hear, say, sound 2, you will see its paired image for 50% of the time. And the remaining sound was um, not predictive at all. So when you hear the third sound, you can see any of the images. Um, and the participants used a small controller to respond. They had two buttons, and if what they were doing while playing the game was, if they think the sound and the image or the picture come from the same pair, they press one button, and if it's not paired, they press the other button. And we recorded all these button presses, how quickly they respond, when they respond, if they're getting a correct response or error, all these behavioral um, effects we recorded. The other thing we recorded was the brain signals. So they had a EEG cap on. It's a non-invasive way to record electrical signals in the brain. So they simply wear a baseball hat or a cricket hat, if we're depending on where you're listening from, and this hat has multiple electrodes beneath it, and what it does is it's a non-invasive procedure. So it just sits on your head, and then it records the different brain signals from your brain. So moving on to what we found was that even though the manipulation we did with the predictability was unaware to the participants, they showed a very interesting behavioral trend. So when they were presented with a highly predictive sound, they were fastest to tell us if the picture really matched with the sound. And they got slower as the predictive value of the sound dropped. So generating slowest response when they were presented with the least predictive sound cue. And it's a very clear behavioral trend, and this was shown in the how this we can see in the reaction time measures, which is how fast they respond. It's a very clear behavioral trend. And in the next step, we really wanted to look at if there are any brain signals that correlate with this behavioral effect. And simply looking at the electrical signals recorded from the brain. And we found that when participants were presented with a highly predictive sound, the activity of the frontal part of the brain would increase, and then it would send a signal back to the posterior back of the brain, activating brain cells that represent the anticipated image. So when you hear a sound, it will send the signal back to the posterior part of the brain, activating the representation of the paired image to it. And what is interesting here is that this happens just after the sound is presented and even before the image is presented. So as if it is anticipating the paired image, our brain is anticipating the paired image. So it's like a priming signal that the frontal part of the brain sends to the posterior part of the brain, which has representation of the paired image. And then eventually when the image is presented to them, because the representations are already pre-activated, they generate a speedier response, as if you're like already little prepared, and then whenever you see the image presented, you are quicker to respond. So all of these behavioral effects, along with the electrical signals recorded from the from the brain, all of this just changed when we administered ketamine. And to just give a flavor of the experiment, what we were doing, and some context to really understand what was going on. So the participants come into lab, and first they play this video game once without any drug on board, and then once that is done, they play the video game once more with the ketamine on board. And this is very minute dose of ketamine, so sub-anesthetic dose, and all the participants are completely awake and aware of their surroundings and of themselves. So very tiny dose of ketamine. And as I was saying, ketamine blocked every single single effect that we saw before. So I would go into the details of what exactly ketamine was doing, but you must be wondering why, what is so special about ketamine, and like what is the deal here. So the answer actually lies in the nature of the connections between different brain areas. Each brain cell, called neurons, actually communicate by passing these chemical signals across tiny gaps between neighboring cells. And each type of signal has a transmitter and a receptor. And what ketamine does is it interferes with one specific communication channel by blocking a certain kind of receptor called NMDA receptors. And this receptors are actually common on the neurons that connect the frontal part of the brain to the posterior part. So the pathways that are connecting these two areas, that's where these receptors are found in abundance. And so when we are administering ketamine, what it is doing possibly is it is blocking this pathway. So the frontal part of the brain has no way to really send this priming signals to the posterior part of the brain, which has representations of the image. So moving on to what actually happened under ketamine was that it generated this very noisy, disorganized activity in the frontal part of the brain. And then when we looked at the connections between the frontal part of the brain and the posterior part of the brain, those connections relatively stayed quiet. So there was very little communication going on between the frontal part and the posterior part of the brain. And then when we looked at the behavioral measures, how quickly are participants responding when they are under ketamine, there was another very surprising finding, is that all the reaction times, that is how quickly they respond, how quickly they press the button, were stretched out. They were slower actually, and there was no difference between the three sounds. So the participants just responded equally equally fast to all the three different sounds. It seems like even though there is this predictive nature in our video game that participants have benefited from before, right now under ketamine there is no priming signal that can really prepare them to make a faster response in for certain sounds. So that priming signal is completely lost. So that is what we found when participants were playing the game under ketamine. But to there is like one small caveat to this, is that we wondered if these effects are general anesthesia effects, and the hypothesis was that maybe if we administer any kind of anesthesia, this is going to happen. And to really look into this and see if this is the case, what we did is we asked the participants to come back for another day, and the series of experiments were exactly same. So they come in, they play the game once without the drug, and then again with with drugs on board. But this time, instead of giving ketamine, we gave them a different anesthetic called dexmedetomidine. And the difference is that this new drug, new anesthetic, doesn't act on those receptors that I was talking about that connect the frontal areas to the back of the brain. And though it has a different mechanism, though this new drug has a different mechanism of anesthetics, but it kind of creates a similar anesthesia state. So, but also this was used on a sub-anesthetic dose state where participants were still awake and aware and can play the video game quite efficiently. And the idea was that there are two possibilities, right? One, participants can show exactly same effects like ketamine, if that every possible effects are blocked. There is no behavioral, there is a no behavioral difference between the three sounds. Or it can be that all the processes are intact under dexmedetomidine compared to ketamine. So what we found was that when we have participants under playing the game under dexmedetomidine, this new drug, new anesthetic, all the behavioral trends, the brain signals, everything was intact. It was as it is. It was before the drug was on board. So whatever effects we were seeing, it was very specific to ketamine and that particular receptor's manipulation. And finally, what is quite interesting here, and I'll be a little bit speculative here because we didn't really test this, but there are some ideas in the field where researchers have suggested that poor predictions are actually poor predictions are symptoms of depression and schizophrenia. So you have depressed patients erroneously predict that the worst can happen to them. And ketamine has been shown to relieve depression-like symptoms in clinical trials. Has it it acts very quickly. It has a much long-term effect. So, and one of the idea is that maybe what ketamine does, it this releases the depressed patients from these associations with negative predictions. So, which is quite in line with what we saw, that since we found that when we administer ketamine, participants cannot really use the existing predictions around your system in that computer video game. And so maybe this is how ketamine works. Maybe what happens is that when you administer ketamine, they are released from those negative associations, and then that is how you can alleviate from the symptoms of depression. And if you are wondering if what is the future direction of this study, so we have collaborators here at University of Wisconsin-Madison, and they have taken up this video game, and they have they have created a long-term study. So they have participants coming in who who are going through a, a clinical trial kind of, and what they're recording is where whether the whether the when this new when these participants play this computer game, whether their behavioral trends change before and after treatment, and they're also recording brain signals to see if those brain signals correlate with the behavioral trends. So yeah, that is basically what I had to talk about today. So and thank you for listening to me, and I would be happy to take any questions if you have.
Yeah, thank you so much. This is such an interesting study. It's really fascinating to me. And since I never did human studies, I'm always fascinated about really good human studies. And so thank you for sharing. And yeah, please flash your mics if you have any questions. Go ahead, Abyss.
Hi, Sonak. Yeah, this is really interesting study, and I think they probably would shed some light on sort of like this predictive association, as you just mentioned, with some of the chronic mental issues like depression. I do have a couple of questions. So the first one is that, do you know, or is there like a floating idea as to how NMDA is specifically targeted as for this specific case. And the reason why I'm asking this that um, like we know that NMDA is actually also involved in a very complex process, like for example, expediting AMPA receptors expression and sort of increasing the intracellular CAM kinase. So, is there sort of a way to kind of target those as opposed to like the NMDA receptor using ketamine to essentially weed out like which mechanism is actually possible? That's my first question. The other one is, do you think it's kind of this kind of results can be replicated using, n- you know, interventions that do not actually involve some kind of drug administration? And by that I mean there are non-invasive techniques like, for example, transmagnetic stimulation that you can actually target with precision as to which part of the brain that you want to um, essentially silence for like in simpler terms. So is there a way for you for your research group to kind of target those parts without using any kind of drug administration to see if the effect still holds using non-invasive techniques? Thank you.
Yeah, those are really great questions. Thank you. So the first one, so we to the reason we are confident about it is targeting NMDA is because there is a dose dependency on which receptor it is gonna work on, and we had a UW anesthetist, and he moved to Sydney right now. But so the dose we targeted from literature, we have found that mostly it is cited to act on NMDA. The AMPA receptor, when it acts on that, it is at much higher dose. And the HCN1, that is one another possibility how ketamine can work, and that was controlled using dexmedetomidine, because the other drug that I was talking about, because dexmedetomidine also works on HCN1 receptor channels. The second one is a great question, yeah, because one of our co-authors, he has moved to Sydney now, but he's actually trying to do this experiment using TMS, where looking at if his lab can really perturb these frontal activations like ketamine does using TMS. So yeah, there might be some non-invasive ways to do it, but yeah, it still needs standardization and probably would need some time before it before we have publication worthy something.
Yeah, thank you.
Yeah, Eric, Nikita, Dennis, please go ahead if you have a question.
I'm still taking it all in, so if anyone else has questions, go for it.
Yeah, I think this study is also, while people are still, thinking, I think the study is also interesting from like a basic, more memory formation standpoint of view. That's why I really like the study where this priming is interrupted, and is really shown how brain regions work together in this priming and prediction. So I think, yeah, I really like that. And what I think it's interesting, there are other studies that try to also address why ketamine is working so well and so fast and so prolonged. But I think this is the only really functional related study. As far as I know, until now, I saw like plasticity and animal model changes that were studied. But I think yours is the only study that goes really into a functional human study. Is that correct?
Yeah, so when we were started to look at this, we were actually quite so the problem has been beat around, but not completely like one story telling the complete picture. So so that was kind of the motive of designing this. This task was actually designed for non-human primates, but happened to this project just happened on the side. So that was also our goal, because we couldn't find something that was telling the complete picture, like what is missing, what is happening when prediction is happening. And another problem we faced or thought of while we were designing is that a lot of the studies use auditory oddball, which is for it's like a train of sounds, and it has some deviation, so it can be low frequency, low frequency, low frequency, then all of a sudden surprise you with a high frequency sound. Um, a lot of studies in predictive processing, as well as how ketamine disrupts predictive processing, is involving that kind of paradigm, like a oddball paradigm. And what those oddball paradigm does is that it assumes predictions. So when you are analyzing a processing of each sound, you are just analyzing the mismatch. You don't know what happened during that prediction period, and so it becomes harder to really separate out a neural element of prediction. And secondly, there is no behavioral proxy or correlate. So so like we didn't find like one single task and study that completely tells the picture. That is also our motivation.
So, what are you planning? So, this was done in healthy adults, right? So, are you planning to do next studies in patients with depression that are naive, I guess, and see if the mechanisms or if the results that are shown are similar? I think that would be interesting to see.
Yeah, so I probably won't analyze the data, but um, we have a group here at the Psychedelic Research Center, and they have a study with the same task, same video game, but not exactly in ketamine. They are using psilocybin, and what they have is the plan is to have patients come in, play the game, and see how the behavioral trends look. Then also look how the fMRI signals look, so the brain activity, and then track them throughout their treatment and see if the behavioral trend changes or the brain signal changes. So the idea is maybe when they're depressed, they won't be able to use the predictive information in the task in the game, and so the reaction times, how fast they respond, are all equal across the three sounds. And as they slowly get better, they start to show the behavioral trends that we are seeing in healthy adults. So that is right now the goal. So we just started this new study. So maybe, I don't know when, but sometime in the future, we definitely will have something out.
Yeah, that is interesting. But so I don't know, but so for the ketamine effect in general, it doesn't really matter what type of priming is happening, right? It's just priming in general is decreased with ketamine, and the theory is that since people with depression have a more negative prediction, that's why the prediction, that's why the depressive antidepressant effect comes in, right?
Yeah, so that is so the so we really cannot pin, like in our study we couldn't really pinpoint where it is breaking down. So there are two possibilities, right? One is whether ketamine, when we introduce ketamine, it is it is creating this negative predictions in the frontal cortex, and then there is also this effect where it really cannot talk back to the posterior part of the brain. And what in our study we couldn't tease apart is who is driving, like which is causal here. Like, is it because the frontal cortex is all messed up when I'm giving ketamine that the talk communication is breaking down? That is probably the most possible explanation, but it's not very we really didn't look at that cleanly. So so the prime, as you were saying, the priming signal breaks down. That is true, that I agree, yeah. But yeah, we couldn't really pinpoint of which of the two mechanisms is driving it.
Yeah, interesting. Because in my in my ideas I had before giving ketamine or any of these drugs was to basically expose. Let's say you would do this with. So, do you? Let me ask first. Do you think that this would also be a good therapy for people with anxiety and PTSD?
Hmm. Uh, I don't have a strong opinion here, but when I think more of about this, it's I what I think can be done with this is it is like their predictions or whatever they're thinking about the world are stuck in one place that is creating negative associations. It's like a solidified ice cube, and then if you give ketamine, it kind of melts everything, and then maybe using cognitive behavioral therapy you can really take the the liquid to another area in the, I don't know, in the realm, and solidify it there where you can make more positive predictions about the world. So but yeah, this is all very hand wavy. This is this might not have any scientific basis, but just some thoughts.
Yeah, but go ahead. But let me just. Yeah, that's what I was thinking. That's what I also always thought. And then, so my idea, or probably a lot of people's idea, is that if you would basically expose in a safe environment people, let's say with PTSD or phobia or so, um, and have ketamine at the same time, or any of these other drugs that have similar effects. Um, yeah, I thought that would maybe, yeah, would maybe help to basically give the brain a chance to rewire. But based on your research, you don't really have to do the exposure part. I think. I think you can just use it in general. Um, I don't know. I'm not sure, but based on your research, I kind of changed my mind to maybe you don't need to use exposure at the same time. You can just go ahead and give ketamine, and it should all predictions should be disrupted. So mostly the negative ones, I would say, in people with PTSD.
Yeah, but that would, exposing them to ketamine would bring it to baseline probably, but how to make sure that they don't fall back there again when they recover from the dose of ketamine? So that's where I kind of I don't know what's a good yeah, how to better guide them to a more positive predicting world. So that's what I was thinking, maybe something like behavioral therapy, and now it's in a more malleable state, and now you can use more reinforcement to really guide this to somewhere where it is gonna stay.
Yeah, thank you. Abyss, go ahead.
Thanks, Kat. I was gonna probably chime in. I think like there's probably a floated idea that ketamine and psilocybin might be kind of ways to kind of disrupt the default mode network, which I believe is sort of the connection between the medial prefrontal cortex and sort of like the posterior cingulate cortex and the angular gyrus. I don't know what the cellular mechanism at this point, but I believe like probably the the outwardly look of people that are mired in like depression, any other kind of like PTSD or probably like suicidal thoughts or um, kind of looped into this default mode network, and the whole theory is if we kick them out, they will probably start to see the world differently. And I think Sonak can probably correct me on this, but this is the impression that I had when I first saw that psilocybin and ketamine can be applied. Um, depending on the dose, obviously, but with the predetermined dosage that they can be used to treat depression and other mental illnesses.
Yeah, I'm not very familiar with the literature here, but yeah, that seems like a possible explanation, that just taking them out of that negative association they had about the world. Yeah, but I don't know how much default mode. So, and my opinion or whatever I'm saying is very much in in the context of the task or the game I designed, because the task incorporates lateral prefrontal cortex for the because of whatever anatomical connections are known between these areas. It is most likely incorporating lateral prefrontal cortex, and then in auditory parabol areas, and superior temporal sulcus, the face cells, because I had this face like shapes, so which are not part of default mode network. So in the context of my task, I'm saying, so maybe I don't know what will I don't know if default mode network is really contributing in any way here, but in a broader context, maybe that is true. And also, I'm not an expert in this area, so that that should be also taken into consideration.
Yeah, we have a few new people on stage. Dennis and AO and Ripley, please go ahead if you have questions.
I had a question and a comment. I will do the comment just very briefly. Depression also as part of other things like bipolar 2 and bipolar 1 are a collection of symptoms, not a disease. Not all of them are due to negative perceptions causing depression. So the end game solutions are going to be very varied. So that is one route, but it's being able to divide things up so that looking at inflammation versus neuroplasticity and depressive thoughts and interrupting that, and so many different things as a part of it. And my question is, one of the things that is often mentioned with ketamine in humans is the NDMA and glutamine effects. sorry, glutamate, and something that is seen really strongly in animal models, at least, is uh, neurogenesis and increased spine formation. So I was curious if you had any information about uh, whether the new anesthesia you are experimenting with also has some similar neurogenesis and spine effects in at least mice models.
Yeah, I'm not really. I know that ketamine literature from some. I have heard about the works from some of the folks here in campus, but yeah, it's we haven't really um, investigated that, so I don't know if something like that is happening, or there is, if it is dose dependent, that also makes it a little complicated, because yeah, we used a dose that was quite low, and um, so I don't have a concrete answer for your question.
Yeah. AO, do you want?
Yeah.
Yeah, go ahead.
I just have a quick question. So, I keep rephrasing this in my head. Hang on. If is this more of like an investigative study that, under the theory, that a certain type of depression and um, maybe some aspects or some people's schizophrenia is caused by poor prediction modeling, and the ketamine is more trying to recognize the channels involved with prediction modeling, and then through association we can say that perhaps reworking those predictive channels could help with depression, um, and other like treatments. And so, because when you recommend behavioral cognitive therapy, like the repetitive actions of altering your thought process is like a more long-term solution, as you're saying. So the question is this more of an investigative study to the channels using ketamine as sort of like the marker, or is this more of a study to see that ketamine would be, in even in low doses, a type of medication for this kind of depression?
Yeah, so we the idea of depression, it's kind of we didn't really design the study to look into depression and this, how ketamine can really help in that symptoms. But the findings of the study, when we tried to interpret it in a more, broader aspect, like how this, what does it tell extra, that's when, looking at the current literature, we thought maybe this is how it fits in. So so the connection between our study and maybe ketamine helping depression in certain, in this way, is in that context, that we were trying to fit our findings and into the current ideas and literature that's in the field. So yeah, we it was not really, like, we didn't really design it to study that.
So it wasn't designed to study ketamine as a medication. It was designed, or it wasn't designed to study depression. It was just designed to study planning, and then using the literature in the field, you were able to say that this could be a possible thing to study later.
Yeah. So as I was saying, some collaborators here have picked up on that and are trying to follow up on that study. or follow up on how um, this kind of predictive processing can be altered. So yeah, so that is that you're correct. Yeah, that is the case.
Cool. Thank you. Yeah. Really interesting.
Yeah, just to give people a short, very short summary for people that arrived later. Sounak is in someone's lab, and he did a study with primates and humans, and basically um, he studied how different brain areas during gaming, like during playing games, make predictions. And then he gave a low dose, or a doctor gave low dose of ketamine, and they saw that these prediction abilities basically were downregulated. And this was really interesting and not seen before. And then they tested if it's just a general anesthesia effect, but it's not. It's quite specific to ketamine. And then, yeah, the sometimes does that these receptors that ketamine target, NMDA receptors, are involved, and they interrupt this communication in the brain that enables the brain to make predictions. And the idea is that then if predictions basically are disrupted, this could be the way ketamine is helping currently people with depression disorders, is that basically this negative prediction um, bias basically gets disrupted by ketamine. Yeah, so just to give a short summary, and if anyone in the audience or so has another question, we'll go maybe around 10 more minutes. And if that's okay with you, so like I just said 10 minutes.
Yeah, that's fine.
Yeah, please go ahead and ask. If not, we'll just keep chatting up here. Yeah, if nobody else has a question, uh, I I think it was quite interesting that that other anesthesias didn't have the same effect, because I thought that when brains are basically under anesthesia, that that basically this is exactly what happens, right? That basically the communication between different brain areas is restricted or gone, and that's why we are basically unconscious. So so you and your explanation was really that the the receptors ketamine target, right? Is that right?
Yeah, that's so when we use that other drug dexmedetomidine, it's a mouthful of name, but um, they were actually more sleepy compared to ketamine, but they were doing the task using those predictive cues, whereas under ketamine, even though they were more awake and aware in physiological parameters like pupil movement, heart rate, etc., they were still unable to use any of those predictive cues. So yeah, um, like it's hard to explain the effects from anesthesia, just simple general anesthesia standpoint.
Yeah, that's very impressive in your study. And how long were those effects? Did you how long after did you test after the ketamine exposure?
So yeah, that depended on the participants, and it was around 3 to 4 hours after we turn off the drug, they will be completely out of any effects of ketamine. That's when we will redo the test again.
Oh, okay. So, are you planning to do maybe to retest maybe a week later or like a month? I think the ketamine treatments are around a few weeks to like the frequency to up to a month. I think if I'm not mistaken. So, are you planning on doing a more long-term test after?
Right now, probably not. Yeah, we our anesthetist moved to Sydney, so we don't have a lab to run these experiments anymore. But yeah, right now we don't have any plans with it, but we have some plans with the psilocybin study.
I thought of another question. I think that it is really fascinating that you chose to use such low doses and got such interesting results. I was wondering how your dosing compared with a similar with treatment levels of ketamine in the short term, and if you know them in the long term, because there are people that receive intravenous ketamine monthly as a maintenance.
Yeah, so we looked at that, and ours is at least half, if not less, than the normal clinical doses of ketamine for treating depression. So very low, and we titrated it because we were tracking the plasma concentration as this whole experiment was going on, um, and we made like, because they had to do the task, we had to make sure they have they are awake and aware, and they have no nystagmus, so their eyes are moving properly. So keeping in all those things in mind, we had to titrate the dose, which ended up being almost at at least half of the normal doses for treatment.
Okay, thanks. Yeah, so I was expecting more of an order of magnitude from the way you were talking about a tenth, but still, yes, interesting to get the effects on a lower dose. Um, let's see. I had a further question, but I forgot it, so maybe someone else has one.
Yeah, I would like. So, it would be also interesting to compare to emotional um, cues if the fact is similar or not. I think that would be interesting if there's a difference to positive versus negative emotion. But yeah, maybe even with other hallucinogens, it would be interesting. If you can So, are you planning to basically repeat the study and yeah, are you planning to repeat the same thing just with hallucinogen? And then I think it would be interesting to do like negative charged emotions versus positive. If the priming basically is also disrupted, or if there's a difference between those two, I think that would be really interesting.
Yeah, that are really good suggestions. We don't have plan to do that with emotional regulation, but um, we have plans to replicate this in non-human primates. So same task, same everything same, just looking at more circuit level with invasive recordings, that what is breaking down, or how is the communication happening, which brain areas are contributing. And we also record from electrocardiographic patients, so medicinally non-treatable epileptic patients, they volunteer sometimes to do these research studies. So we also have this, so it's like a same task across multiple species and multiple techniques. So right now those two are probably the priorities, and um, but we haven't really thought about, yeah, that's a good point about emotional regulation. So yeah, we haven't thought about it, but these are very good suggestions.
Yeah, I don't know, at least in animal models, I did my PhD in fear memory in rats and mice and Joseph Ledoux's lab, and um, usually the fear, at least the fear related ones, can in subjects, like even in rats and so on, there are individual differences, but become quite unflexible, and also habitual avoidance of like a threat can become very is very unflexible. So it would be interesting to see to see differences in this type of memories, and memory priming, if it can disrupt anything, or it clinically, translationally, it would be very interesting and very important, maybe. So would be really cool. And yeah, thank you.
Yeah, that is true actually. Now, if I am thinking more about it, it has a lot of translational value. I agree.
Yeah, like especially habitual avoidance. I don't know. Also for getting grants and stuff, it's like pretty good to get grants, because it has like implications for addiction, for compulsion, for all kinds of stuff that is problematic to address. Because, but yeah, it would be also from a basic science point of view to see differences in these memories, and maybe there are different mechanisms involved that maybe ketamine can disrupt or maybe not, and why would be interesting to see. So it's very cool. Your study opens up a lot of new questions and ways to go, I think.
Yeah, yeah, there are some ideas that hippocampus probably contributes. So once they learn the associations, like how is the frontal cortex bringing them into this, like there are like two steps, right? First you need the associations, and then you have to overlay the probabilities on top of it, and maybe prefrontal cortex is doing the probabilities, but how is, they need to bring the memories from somewhere, and somewhere there probably all these different contributions of memory come into play, and hippocampus and say entorhinal subiculum, all this yeah, and these have connections with prefrontal cortex, at least in monkeys that I know of. And so there are some there is one paper on it, but there are some ideas. So one cool thing about recording from electrocardiographic patients is that because we get a lot of medial temporal lobe epilepsy patients, they have these electrodes in all those medial temporal lobe areas, hippocampus, amygdala, um, some entorhinal electrodes too, but a lot of hippocampus and amygdala. So we are looking into it, but we won't get a chance to really perturb the system with ketamine, but we'll have probably a more mechanistic view of how is this different subsystems, say the memory system or the more frontal networks contributing, and maybe how, I don't know, thalamus is really manipulating, um, like a ringmaster, like taking information from one area, put connecting it to another. So yeah, it is still up in, like it is, we are still collecting data. So
Yeah, so why I'm saying this you know, my PhD supervisor, Professor Joseph Ledoux, he wrote a whole article and basically a book about it, why we didn't make any progress in addressing like anxiety disorders and PTSD and so on, is because in the animal models we use, um, that have basically weaker prefrontal cortex and so on, like it has a less weight on the system compared to humans, and because we did all these studies on animals, uh, in animals, the drugs we discovered for it work pretty well, but they don't really address, they don't work well in humans. They kind of suppress like the whole activity of a human, but really anxiety uh, is really not gone, because he says um, the weight is for sure bigger and higher cognition areas in humans, especially prefrontal cortex and so on. So actually, his lab and colleagues of mine are, more and more focusing even in animal models on prefrontal cortex now and so on. So that's why I think it it would be really interesting and important to address this. Like Professor Joseph, he wrote like a whole article that people were upset, but he might be right that we focused on the amygdala and all these older brain regions too much, because we in the animal models we use, they just have such a big weight, but it's not really working for treating humans. So I think that's why your work is really important. And yeah, congratulations. And yeah, please keep updating us with your research. And yeah, I wish you all the best for your PhD dissertation. I know you will do great. And thank you so much.
Thank you, thank you, thank you for the invite. It was nice talking here.
Yeah, Abyss, go ahead.
No, I was just gonna say thank you for taking your time to share your research, and I hope you'll join us soon with even better result, like a more really interesting results as well. So yeah, just wanted to mention that. Thank you.
Thank you.
Yeah, come back. We'll have yeah, and everyone currently in the room. We have tomorrow. We have more guest speakers coming. So tomorrow we have Dr. Detlef Weigel from Germany. He's talking about his more evolutionary biology studies, where he shows that mutations are not random after all, and they are quite biased, which kind of changes or shakes the theories we have about evolution currently. It's a really big deal study. So he's here tomorrow, 1 p.m. EST. And then we have let me check the time. We have on Thursday at also 1 p.m. EST, Dr. Kalsa. He's a colleague of Dr. Olu, and where he shows that anxiety is associated with autonomic hypersensitivity. So it's also a really, he's a really great scientist, and yeah, he will take the time to talk with us. And then Friday we will have Dr. David, and she did a study where she gave repeated low doses of LSD to healthy adults, and she will share the study results with us. So yeah, stay tuned. Come back, maybe Sunak. Maybe there's some. I think there's some stuff that is also interesting for you. Um, the cool thing here is that people just get to interact with with real scientists doing real great interesting work currently. So yeah, thank you so much everyone. Check out the press on the Science Society, follow so you get alerts when we have great rooms like these. And have a good night, good morning, good day, wherever you are around the world. And see you again soon, or hear you again soon. Thank you.
Thank
Bye.
you. Bye.
Bye, everyone.
Take care, everybody.